Sitting in the seminar room of one of the oldest private research institutions. Carnegie Institution was established in 1902. Certainly one of the oldest, if not the oldest, private research institution. We don't have education unlike many other universities. Our mission is basic research, but we do care about education and training. And for us, this program has been running for many years, every summer. We have very enthusiastic postdocs and professors engaged in this summer intern program. I see more people coming in. Maybe I'll hold a little bit. It's kind of never walking here. All right, welcome to the summer intern program of Carnegie Institution. And this is a program we have been running for many years. And traditionally, mostly plant biology together with Stanford plant biology colleagues. And more we have our global ecology colleagues joining us. And again, about the Carnegie Institution, we have 2 of the 6 departments here. Of course, now in recent couple of years, we reorganized into 3 divisions. So we are 2 departments under the division of biosphere science and engineering. Therefore, the division also includes not only these 2 departments here on Stanford campus, but also our embryology department, which is currently at Johns Hopkins University campus. So that's the overall organization of our institution. But the research here focuses on plant biology in our department, global ecology. Together, we basically try to understand how plants in the ecosystem shape the ecosystem and how plants are shaped and adapted to, change in the environment. That's kind of the focus of the research here. So this program gives you an opportunity to really experience basic research, and you get a chance to hear about the science today from 2 of our colleagues, Devaki Bhaya from plant biology and Lina Richmond from global ecology department. So I really hope that you will take the opportunity to, learn through this summer. I know you will be doing research in the lab with our researchers in the lab, and this activity every Friday will give you a chance to hear from prominent scientists in the world about their research. And some will be from our outstanding postdocs, I know, to talk about their specific project. So I will encourage you to engage, and again, learning is all about engagement. So be brave to ask some questions, any questions, no questions too stupid to ask. So I encourage you to ask questions and engage in the program. I know in the end, we'll have a poster session one day at the end of your program, and everyone will make a poster, and that's another opportunity for you to really talk about your own experience and research. So for many of you, hopefully this will be one of the first steps into a lifelong career in research, and maybe in plant biology or biology related. So enjoy the experience, and I wish you all good luck. Excited to see you all here, and I'll hand over. I don't know who's going to speak first. Elena or Devaki, you can take over. Again, welcome to Carnegie. I'll be around on campus, so feel free to stop me if you have questions for me.
Hey, hi everyone. Can you see my slides?
Yes, we did.
Okay.
But it's a presenter's view.
Okay. All right. Hang on one sec. Is that better?
Oh,
yes.
Yeah?
Yes.
All right. So here we go. I'm really sorry I'm not there in person, but I wanted to start by saying that this is what Carnegie really looks like. It's full of colorful people, and I really hope we'll get a chance to talk more in person. But this is just a postdoc meeting that was held just about a couple of weeks ago. And you can see that there's really a lot of people doing very interesting work. And I'll leave it at that and go on to my next slide, which is to emphasize what I think Jiyoung said a little bit, and that is that all of us as postdocs and as faculty have many, many hats that we wear. One hat that I love to wear is to teach. And I've been teaching at Stanford for many years. One of the courses that I taught at one time was called Face Bug, The Secret Life of Microbes. And I just finished teaching a class called Partner with Trees. And it was a lot of fun. I should say that I'm a molecular biologist and a microbiologist by training, but I've always loved trees. And this was an attempt to get undergraduates really interested in looking at the trees on campus. And I want to offer the opportunity for any of you who are interested to take campus walks around. On the left of this picture is a picture of me with somebody who's a very well-known author. She came to Jasper Ridge, which is a research site that Stanford has. And she was talking about native knowledge and trees, and it was absolutely fascinating. So I want to move on to tell you a little bit about myself, just because it is an opportunity to share our histories. Like many of you, I'm sure you got interested in biology because it has so many surprises, the way flowers know when to bloom. The second picture is a fascinating experiment that was done by my school teacher back in Calcutta, where she actually took a cow's eye, sounds a little gruesome, and she put a candle in front of it, and she showed that at the back, there was actually an inverted image of the candle, and showing that there was a lens in the eye. And I thought it was fascinating, and I've continued to be an experimental biologist. I've also come very many places. I taught for many years at the university in Delhi, and I've been at Carnegie for many years. I took a 2-year break to go to the National Science Foundation. Unfortunately, I can't see you guys, so I would have loved to ask how many of you know about the National Science Foundation. But it is a science foundation for all of the US that funds both basic science and education. So it is remarkable in the span of things it does. And if any of you are interested, I'm happy to tell you more. So what I want to talk about today is about microbes. And I know Jiyoung has called this a department of plant biology, but I would argue that plants are practically microbes in the sense that the green, the beautiful oak trees you see in this picture are actually green because they have chloroplasts, and the origin of chloroplasts are bacteria, cyanobacteria. And the numbers down here tell you another picture, and that is that we are far outnumbered. You can see the number of humans on the planet, approximately 10 to the 10. Number of bacteria, 10 to the power 30. That's pretty impressive. You have 10 times more viruses than that. And we know very, very little about them. That is take-home message number one. Take-home message number two is from Rita Colwell, who is the director of the National Science Foundation. And she said that really the natural history of life on Earth is exclusively the history of microbial communities. And I tend to agree with her. That might be a bit of a stretch, but I do think that it's important to realize that what we don't see can be just as important as what we see. And so for much of my career, I have focused on bacteria that are called cyanobacteria, and cyan sort of means blue-green, which refers to the very lovely color that they have. And the reason that I've been fascinated by them is that they are really an ancient group of cyanobacteria, and I'll come to that in the next slide. What's fascinating about them is that they can fix carbon, they can fix nitrogen, they store phosphate. So they're like a treasure trove for anybody. They really do the basic biology that keeps this earth in motion and functioning. They're also amazingly resilient. You can dry them out and they come back. You find them in deserts, in the desert crust. You find them in the Arctic. They have gone everywhere over a very long time. So that in itself is interesting. But because of that, because of the fact that they do photosynthesis, you can also use them as biofertilizers, biofuels, pharmacological compounds, etc. And what I want to also emphasize that in the last 15 or 20 years, because genomics and omics technologies have exploded from just being observing nature and looking at physiology, we can now go to looking at the genomes of these organisms. And that's what I want to tell you more about today, the approaches we've taken. But here's a fun little slide that I found, and the link is down below if any of you wanna have it for later. But it shows the history of the Earth simply as time travel. If you started in LA and moved to New York, and you can see here that I've taken a screenshot of where cyanobacteria emerged, which was an amazing 2.7 billion years ago. And they've basically hung out for all that time here. So that alone, I think, is a staggering feature. The Earth started in LA, right, about 4.5 billion years ago. And so while you ponder that, I'll also show you something else if it does play. Hopefully, I'm not sure I'm going to be able to play this. Maybe I can show it to you later. It's really something else that I wanted to bring into this that I've been thinking a lot about, and that is trying to bring other features of my interest into science. One of them is art. And what this actually shows is, let's see, maybe it will play. Here we go.
I don't hear anything, Devaki. Should there be sound? Devaki, we don't hear anything.
Sorry?
We don't hear any sound. Should there be sound?
It's not that important, but I mean, maybe I'll just move on to the, oh, sorry. How do I get out of this now? Can someone help me out there? How do I get back into, okay, watch later. Huh.
Try escape, Devaki. Hit escape on your.
Yeah, I am. I have. Yeah, I'm trying. That doesn't seem to do it. Can I get out of here?
Unshare your screen and share again.
Yeah, hang on. I have some expert help here. Just share my screen with the PowerPoint. Yeah. Yeah, but get out of that one. No, that's what I don't want. All right. Does that look good?
No, we don't see you.
Hang on, one sec.
Your screen.
That's it. Okay.
How is the present? Okay.
So you have to just go into the. Is that good?
Yes. No.
So we see the presenter notes, FYI.
Okay. Just that one. Yeah. And you just go to display.
There we go. Perfect.
All right. That experiment was partially successful. Can everybody see now? All right. So what I want to say is that despite what I was trying to show you, which was really a small clip of how photosynthetic microbes behave, generally they are all for the good of the earth. But I also want to bring out the fact that because we are in a state where the globe is changing, we have things called harmful algal blooms, which I'm sure Elena will also be talking about. We have things like pollution, the needs of energy, we have uncontrolled fires now. So a lot is going on in the world, and I like to call it catastrophes without borders, because they don't respect global borders, they don't respect state borders. And so we really have to pay attention to the fact that much is happening in our world, and how do we deal with it? Now, the way I've tried to deal with it is to look at phenomena that are happening in our world that we can look at. One of them that I'm particularly interested in is the behavior of microbes, and then linking that to microbial ecology and to the genomes. So in a nutshell, that is what we're trying to do. And using that information, and we'll be talking about that later, to build synthetic communities. Now, these are the areas that we've been talking about a lot in the lab for many years. That is, first, trying to understand collective behavior of microbes. And the phenomenon we've looked at, and Freddie Bunbury later on in part of these talks will be talking about it, a phenomenon called phototaxis, where microbes can actually see light and move in that direction. I'm also very interested in phototrophic communities, and that's what I'll focus the rest of this talk on. And that leads to this idea that I think will really have a great impact on microbiology and synthetic biology. And that is, rather than look at microbes as individuals all doing the same thing, we know that they live in communities. So to try and understand how they behave and whether we can then start to use them in intelligent ways is, I think, some of the things we want to start thinking about very seriously in the lab. So just very quickly to show you the phenomenon. So you see a yellow arrow where light is coming in, and you see groups of cyanobacteria move. And this was really to show, because at the bottom right is not the cells themselves, but a simulation of that movement, which really says something that I'm fascinated by, and that is, can you take behaviors and start to model them in ways that you work with mathematicians or with physicists? And the point being that if you can make these models, you can also predict behavior. Once you can predict behavior, you can start to change things in organisms. And that's the sort of arc of information that we want to use. So I want to tell you a little bit about the work we've been doing in Yellowstone National Park. And many of you know that it's the first national park that was declared in the US. Several of these are just nice pictures. We work in a place called the Lower Geyser Basin. Now, the real big message I want to leave you with, even if you don't get into the nitty-gritty, is I think of these environments as being giant outdoor labs where you can do a lot of experiments, because you have a situation where microbes have been living there for millions of years. You have a stable temperature gradient, and as the water flows out of these, you get microbes, and you can start querying them in very detailed ways. You can look at the light levels as they change. You can look at levels of gases, and all of this is happening in an environment that is both fluctuating as well as stable. And these extremophiles, like cyanobacteria, have evolved to deal with this. And here's somebody, Sheila, who was a graduate student many years ago in Michael Kuh's lab. And you can see that you can take all of this instrumentation in there. And I won't bother with giving you too much of this detail, but it really tells you all the advantages of using the natural environment and national parks to do this. One of the things I would see, the last one on this, is that many folks think, including the folks at NASA, that some of these extremophile environments could be a proxy for early life. So As you well know, there's a great deal of interest in trying to figure things like that out. Was it an RNA world? Was it a DNA world? And I think these extreme environments give you a possibility of understanding that. So how do we actually do this? So this is to give you a very bird's eye view of what goes on. You can start at the left, for example, you get a sample out of the maths, right? And that itself, of course, is not trivial because you need permission to take anything out of the math. You can grind up that top green layer that you see, and you get something that we call the metagenome, because you can get, if you do it right, all the DNA out of these samples. You can also enrich, for example, the next slide shows you the cyanobacteria, which are highly autofluorescent. That's why they look red. And then you can take individuals and you can get their genomes. And what we've tried to do in the lab over several years, and we're now actually beginning to succeed, is to grow these guys in the lab. And what's the advantage of that? The advantage of that is that then you can start to get much more specific about the questions you ask, because you can build a genetic system. You can look at synthetic communities, and you can also look at evolution. It's and because these guys divide so quickly, they're not like plants that might take 6 months or several years to divide. They're dividing on the order of hours or days. So you can actually do evolution in the lab. Now, to step back just a little bit and give you a sense of what you need as a backdrop to this, we do have enormous amounts of information already at hand. One of them is what's called a phylogeny of relatedness. So what you're looking at here in this kind of tree-like structure are various cyanobacteria, and you don't need to remember the names. Just look at the blue and the red. So those are two of the enrichment cultures that we got, something called OSB and something called OSA. So one has adapted to growing at 70 degrees, which is about the temperature optimal for photosynthesis. You don't really get photosynthesis, but at 70 degrees, I might add that if you put your hand in the water, you get scalded. Yet these cyanobacteria are able to survive and do photosynthesis. And then you have OSB, which is at the lower temperature, about 50 degrees. And so our question starting up several years ago is, what makes these two guys different? Why can one survive at 70 degrees? The other can only survive at 50 and can't survive at 70. Can we get at answers? And those are of fundamental importance as well as interest to us. So I'll leave you just with one example of what we did find and what we call comparative genomics, which is an enormously powerful way to look at the differences between organisms. So here you have a circular genome. It's about 3 megabases. So you have Synechococcus on the outside, that's OSB. And now the big surprise to us was that if you look at these two genomes and compare them, they don't look similar in organization. In other words, to step back just a minute or two, if you have two organisms that are very, very similar, usually their genes are very similar and the organization along the genome is also very similar. And what this tells you, without my going into the details for something like this, is that there has been a lot of shuffling of these genomes. And this is taking just two individuals and comparing that. And what it's telling you is that for some reason, and we're beginning to get those answers, these genomes are highly shuffled. And the shuffling has many implications. One of them I hope you're already sort of thinking about is, what's the implication of the shuffling? It means that if you're looking at enormous populations of these cells, it's possible that every individual is slightly different than the other individual. And I hope that makes some of you think that bacteria are not guys who are all the same. They do have differences, and because of these differences, selection can act on them and evolution proceeds. So what I want to leave you with this picture, with this very simple idea, is that what's happening is a very dynamic population. And we are looking at it, in this case, just through the lens of these two genomes. But what you also can do, and again, I won't, belabor the point, because I hope some of you will come by and talk to us, You don't need to look just at genomes, which takes a lot of work and a lot of money, although it's getting easier. You can also, remember I said you can get everybody from the environment, which is what's called a metagenome. But once you get a metagenome, and the analogy holds that if you have a library, it's all organized by books. But if I tear all of the books up and then say, well, can you reorganize this and tell me which page came from one? It's a very difficult computational task. But that's what we're trying to do with the genomes that we have from the hot springs, and trying to assign them to different organisms. The point being that without having isolated them, we can still understand everybody who's there. And once we know who's there, we can then begin to ask, well, what makes them different? How do they interact? And that is something that Amanda Shelton, who's a postdoc in the lab, will also be telling you in one of these talks. So that's to give you, sort of bird's eye view of all the kinds of questions that we are pursuing in the lab using a number of different techniques. But at rock bottom, the question that fascinates me is, how do thermophilic communities adapt to life at these very high temperatures? And if we begin to disentangle some of those, we will begin to understand, I think, some basic principles of how photosynthetic microbes have actually evolved over billions of years and are continuing to evolve. So I'll leave you with the pictures of some of the folks. Hopefully you'll come visit us. One of the great joys of being at Carnegie is that we can collaborate with many, many folks. And I hope you will, as interns, take that spirit of collaborations. Come, you don't have to stay in your own lab. Come visit. Here, yeah, I'm showing you Amanda and Freddie who will be giving talks, and I'm sure you'll meet Carlos and Evan, Vicky, several others. So I want to leave you with this picture of Carnegie since I can't be there. But I'm sure if you wander around, you'll see the famous hammock where you're welcome to take a nap and dream. We often do science outside, or have in the past. There are places to sit and chat, and there are always parties. So it is a great place to do science, and it's really up to you to utilize all the advantages we do have at Carnegie. And so I'll leave you with one question, since I'm not there. And I've given you a flavor of all the kinds of things we can do. I'd love to hear if any of you have thoughts about what experiment you would do if you had access to these incredible environments. And I'm happy to talk to you more about that, even offer some prizes. So I'll end there. That's some information about me and talks I've given in the past. And I'm happy to take questions. So I'll probably stop sharing so that becomes a little easier to talk. So I'll escape out of there. Okay. All right, so there's a question in chat, but I don't know if somebody's moderating. How do you want me to do this?
Feel free to read the questions out of the chat, but I'd like to start with interns in the room. So
okay, sure.
In the room, let's start with those.
Questions from the interns?
No? Okay, maybe I'll get the wheels churning by asking a question from Jason. He says, generally speaking, how old are these microbial mats? For example, how old is the core sample in the slide that I showed? What time span are we looking at? So that's a fascinating question. And I'm not sure I can give a precise answer. What I could say is that these must be tens, if not hundreds of years old, the pieces that I'm looking at. But one thing I should mention is that very often these mats are torn up by either hailstorm or bison stepping in them, and then the mat has to reorganize. And what's an interesting question to think about is what are the principles by which this mat reorganizes. How do the cyanobacteria come to the top? How do these other guys who are also photosynthetic in some cases, how do they reorganize in a mat? And so another point to make, Jason, is that the mat is growing mainly by the cyanobacteria at the very top. So they need the light. And as they die, potentially, maybe they're being pushed to the bottom. It's one of the questions that is really very intriguing and a kind of question, in my opinion, we should be trying to answer in the lab by building up these synthetic communities.
Question, audience here?
Yeah, I have a question like on a much global scale. These environments that are extreme are kind of separate in space. And do we see that there is a community that is different for each of these niche environments around the globe, or is there somehow there happen to be transfer of these organisms specialized for hot springs that have colonized all over the place, and what's the view about how they've been traveling around?
Yeah, so that's an excellent question, because as you know, and there are beautiful maps of that, there are volcanic and thermal features all over the world, and along regions. And in those, wherever there's hot springs, depending on the environment, so if you have an alkaline environment, for example, you very often get cyanobacteria. Very often they are actually unicellular cyanobacteria, but not exclusively. So the question really is, how do they travel around the globe, right? And we do know that cyanobacteria can be aerosolized and move around. Now, whether that's the way it's actually happened, we don't know. What I would argue, and I would love to do, in fact, I was just talking to someone about it, is it would be great to get samples from 20 different hot springs around the world and then do this kind of metagenomic, I see Elena's nodding, to do this kind of experiment to answer exactly that. And you can do it with hot springs that are very close, like, 2 hot springs in Iceland, and then do one between Iceland and New Zealand and see how different they really are. And it was a question that has obviously been asked before, but mainly at the level of 16S phylogenies, which I don't think tell you the entire story. Just like I told you, because there's so much shuffling going on and horizontal gene transfer, you do get some idea, but not the kind of detail that I think tells you what allows hot springs to harbor these kind of environments. And I should also mention that, right next to the springs that we worked in, there are acidic hot springs, which have a pH of 2. And there are bacteria going there, not cyanobacteria. So you do have the environment playing a very strong effect on who comes up in terms of what you expect to see in different extreme environments. Hope that answers your question, Solen.
Yeah. As you have mentioned that there will be a gradient of temperature in the hot springs.
Mm-hmm.
Will that remain same? Because I have a doubt, like there will be a change in the seasons. Throughout the year, there will be the change in the season, there will be change in temperature and environment.
If I understood you correctly, what you were asking is there temperature variations as a function of the season?
Yeah.
So in fact, amazingly not. And that is actually one of the things that make it very special, because the temperature doesn't change over the seasons, but the length of day does. And as you probably know, cyanobacteria have circadian clocks. So they do function in terms of the circadian rhythm, but the temperature stays more or less static. So I just got a message from Elena that we should wrap up here, but hopefully from next week I'll be around, and everybody from the lab is always around, and we're happy to answer questions. So welcome again, everybody.
Thank you so much. We're going to pass to our next speaker now, Elena.
All right. Hi, everyone. So I'm sharing my screen, and please let me know if it works. Yeah, do people see my screen?
Yes.
Okay, excellent. Thank you. So